name is Mr. Shin. I'll be your instructor for EpiBio Winter Bootcamp. So this, as you have been advertised prior, I'll be mostly focusing on problem solving aspect of this course. So I'll be teaching you how to approach a problem more effectively, efficiently, and how to tackle problems using the trick that I'm gonna be teaching you. Okay. So in the next slide, I'll be going over how the class is gonna look like. Okay. So you have this class is, like I said before, a problem-solving focused course. So it'll be a very fast pace, but don't worry. I'll be posting all the relevant materials as if I'm actually running the full course in ahead of time. So you'll have full access to see those materials ahead of time. So I know some of your school probably have, I don't know, halfway or midway or almost finished. So I don't know how fast your school are. It doesn't matter. So I will upload each of chapters with full practice problem, chapter by chapter. So you will have full access to all lecture materials, okay? But since this is a problem solving course, I'll be prioritizing going over the problems and lecture slides rather than the lecture slides, okay? But that doesn't mean I'll neglect the lecture slides. I'll be giving you like a brief overview of class material. For example, if I'm assigning you about membranes, I'll be covering you key concept, at least like last hour or 30 minutes of the class so that you are well aware of the class material. I'm not going to just throw you guys out there, do this by this day. I'm not just expecting you to fully digest the material yet, okay? So what I'm saying is that I'll be guiding you like the things that you need to know for the worksheets, okay? So you don't have to worry about that. And like I said here, this program will emphasize more on genetics and biochemistry, okay? So the reason is that a lot of students have come up to me in the past and they were super confused on genetics and biochemistry part. So I'll be more emphasizing on those aspects, but I'll still be covering all the chapter materials from first chapter to last chapter, okay? So don't worry about that. So homework will be assigned at the end of the class in Google Drive. So I will post all the materials ahead of time and I'll be like, okay, do multiple choice and free response for this chapter, then next chapter. And you guys just have to do it by designated date, okay? And then typical class breakdown is like, assuming there's 2-hour class, first 30-minute to hour will be reviewing the wrong homework questions. So that will be provided by you guys. If you say I got 5 and 7 wrong, then we'll go over 5, 7. We'll go around the student. I'll be getting numbers that you guys got wrong, and I'll be covering those. And then after that, and I'll be covering the new chapter materials. Okay. All right. So more detailed class material will be provided at the beginning of the class. Beginning of the winter bootcamp starts. Okay. So this is just like a demo class, how the class is gonna look like. So I have a lot of slides, and then it'll be looking something like this. Okay. So this will resemble the worksheet problem that you have before. So this is so that everyone can see the screen all at the same time, question by question. Okay. With that said, let's just dive into those questions here. Okay. So these problems or worksheet problems are taken from the membrane, a membrane chapter of the AP Bio. Okay. All right, let's see. So number 1, living things regulate their internal environments. What is this process called? A, selective permeability, B, plasmolysis, and C, upregulation, and D, maintenance of homeostasis. Okay. So when you see this problem here, my instinction kind of thinks that A could be the answer. Oops. A could be the answer. Right? Selective kind of sounds like it can regulate, right? So let's just put like a little like pause here, okay? Maybe the answer may not be the case. So the definition is ability of the cell membrane to control the flow of substance in and out of the cell. Okay, let's just give it a little pause there. B, contracting water out of the cells. So a lot of plant cells use vacuoles to pump in and out of the water to maintain their pressure or concentration. And C here, an increased number of receptors on surface of the target cell, making cells more sensitive to hormones or other agents. Okay, and then we have maintenance of homeostasis here. Okay, so when I looked at this problem, it needs to regulate, right? So regulate means to control at some type of degree, correct? And their internal environments, right? The focus is regulate internal environments, okay? So that sounds more like homeostasis here, isn't it? So homeostasis is that we have to keep maintaining our, for example, body temperature, our sugar level, or things like that, right? So the constant process of controlling using either negative feedback or positive feedback, that would be the case, this case, okay? So living things regulate their internal environments. The key point is to regulate, okay? So only maintaining regulation or maintaining homeostasis will actually fit more for this problem, okay? Selective permeability, I don't think they have that much of a control, okay? So more regulation, I'm leaning towards more D, okay? So that's why I picked D as this answer here. Okay, so I lean towards more D because there was more control or regulate their internal environments here. Okay. Number 2, cell membranes are blank and regulate materials moving into and out of the cell to maintain equilibrium. So I kind of briefly introduced this concept in the previous problem, right? But before moving forward, let's actually look at this diagram or picture I pasted here. Okay, so cell membranes blank regulate materials moving in and out of the cells to maintain equilibrium. That's correct. But there's 2 types of transport that's happening, correct? In the cell membrane. The one that uses concentration gradient, which is passive transport. The one that goes against the concentration gradient, which is active transport here, correct? Okay. So since we don't really have to talk about transportation here, let's just revisit the answer choices here again. So A, completely permeable, ionic permeable, selective permeable, and protein permeable. Okay. So the keyword here is, again, is to regulate, correct? So the one that can regulate is selectively permeable. The word is self-regulate, also resembles selectively permeable. Is that clear? Right? So, which is true, right? Cell membranes are made with phospholipid bilayer here, correct? And it can only select, for example, sugar, water, or carbon dioxide. You can only select those things. You can push through using diffusion or facilitated diffusions here, correct? So with that said, cell membranes are A, not completely permeable, right? That's not true, right? We only let in only selected materials, okay? So ionically permeable membranes are actually both ionically and other like molecule can also go through. So that's not the case. So I think more inclusive term will be C, selectively permeable, and that would be the answer for this case. Okay. So number 3, when we determine the direction and rate of solute movement, we must consider the blank both outside and inside of the cell, as well as magnitude of their concentration gradient. Okay, so let's read one more time. When we determine the direction and rate of solute movement, we must consider blank both inside and outside of the cell, as well as magnitude of their concentration gradient. Okay, let's see. So I posted this picture here. And you can see that chlorine, there is four ions outside. And then you have two here, right? So you have a greater concentration gradient developed outside than inside, correct? So you have green moving towards inside here, correct? And you have also sodium. There's more sodium outside here, ion. So it'll move to inside, correct? And you have more potassium inside of the cell, you'll move outside. And then they will move, each ion will move differently, correct? It's not like they're clumped together, they move all together, correct? Right? So with that said, let's go back to the answer key here, okay? So A, the solution concentration of solutes added together, that's not true, right? Each of them move independently. Concentration of the water, we're focusing about the solute movement, correct? So we don't really know about the concentration of water right now. And C, concentrations of each solute independently. That's true, right? Based on the each concentration here, they move individually, correct? So C would be the case. And D, concentration of ions only. That's not relevant for this case. Okay, that's why C is the answer choice here. Okay, so number 5, water will diffuse in and out of the cell, which allows it to maintain A, equanimity, B, concentration gradient, C, tonic equilibrium, and D, concentration gradient here. Concentration difference. Before we move on, I just pasted this diagram from website. So we have a selective permeable membrane here. You can only move water. As you can see, there is particles, solute particles remain the same amount. As the time goes by, what happened here? You want to maintain, water is only allowed to move between this environment here. And then it has a tendency to keep the same concentration. So this is more dilute solution. This is more concentrated solution. The only way you can keep this solution equal to each other is if water molecule went from left to the right. Is that clear? Right? So we call this tonic equilibrium. Okay? So let's have these 3 conditions here. You guys may have heard this or may not have heard this. So we have 3 types of condition here. We have hypertonic, we have isotonic, we have hypotonic conditions. So hypotonic is when outside has low concentration, right? So to make this concentration high, water has to be sucked in, correct? On the other hand, if there's high concentration outside here, water has to move out of the cell to dilute this guy down to lower concentration. Is that clear? So water will diffuse in and out to maintain what? The tonic equilibrium will be the right choice for this case. Okay? Okay, so number 6 here. Using the illustration, which of the statement below correctly identifies the net movement of oxygen and carbon dioxide molecules? So let's count them up. So we have 2 oxygen molecules inside and 3 carbon dioxide molecules inside. Outside, we have 4 oxygen molecules here and 1 carbon dioxide molecules, correct? So molecules, so from couple slides before, each solutes will move independently, correct? So it's pretty easy to predict from here, right? You have 4 oxygen molecules outside to 4 to 2, correct? High to low here. And you have 3 molecules inside to outside, low, high to low again. So if you find that answer choice, it'll be A. So what is the major difference between carrier proteins and channel proteins? Okay. So a lot of students know what they are. They reside in the channel membrane, membrane. However, a lot of students don't know their distinctions here. So the answer is A, but before I go over the choices, let me just put this diagram out here. So as you can see here, channel protein, carrier protein, they look different, correct? So channel protein, they don't change the shape at all, whereas carrier protein does change the size, correct? And looks like just by intuition, the fact that they have to close it one side to transport the other side kind of infers that these molecules tend to be bigger than protein. Just having that in mind, let's go ahead and lay out the characteristics here. So carrier proteins transport molecules, okay? And they flip between two conformations. That means they physically change the shape and they have low transport rates. That's because they're more used to transporting a larger elements, larger like molecules. So they cannot transport fast. If they transport fast, then they may run into some issues because they're larger molecules. So it'll be difficult. If you let all at once, I think the cell will have issues doing that way. And then you have solute molecules are bound to one side, released from the other side. So let's go to the protein channel here. So channel proteins transport ions, which are a lot smaller than carrier protein. I mean, it's a lot smaller than what carrier protein transports. The position is fixed, and you have high transport rates here. Okay. Solute molecules diffuse through pores, and then, for example, sodium potassium pump. So the reason why this one is more advantageous is that, so we have when you like contract muscles or when you communicate between neurons, the rate has to be super fast, right? Otherwise, you won't react or contract muscle at fast enough, right? So this left side protein channel is more advantageous for faster reaction required element, correct? For example, like reacting through, let's just say you have a ball coming at you, you need to dodge that ball. If your reaction rate is not fast enough, you'll get hit, right? So that's why this left side protein channel specializes in faster communicating cell functions, whereas carrier protein is more so focused in transporting material inside and out. Make sure the larger molecules are transferred in and out. So, for example, a glucose can be an example. Glucose molecules are a lot larger, and they would use this carrier protein to transport in and out of the cells. Okay, so next example here. A fungal cell is placed in a solution of 1.0 mole of glucose, which is highly concentrated. What is the most likely mechanism of membrane transport of movement of glucose into the fungal shell? Okay, I just talked about this, right? Before that, I just have a little picture to help you understand. So we have a glucose molecule outside floating around, and the inside we have this. So we're utilizing this protein channel to take this one and then change shape, and then you have you put this glucose molecule inside like this, correct? So like I said before, it'll use the carrier protein. Is it clear? So in those choices here, not simple diffusion because it uses the protein, right? So facilitated diffusion by its meaning is that we use facilitated help, like a protein channel, right? And then active transport, I don't think we're using any energy in the diffusion process. Diffusion itself means it's by using the concentration gradient. So active transport won't actually fit this case, and excitosis will be using a vesicle to transport in and out of the cell. So I don't think this is the case here. Okay, so you are having high to low, you see, highly concentrated to inside of the cell, right? So this is high to low. So regular type of diffusion case from high concentration gradient to low concentration gradient. So it has to be diffusion, and it's either A or B. And then A cannot be the case because glucose molecule is large, correct? So the most appropriate answer will be B. Okay. All right. And then we have a problem here. So plant must uptake mineral ions from the surrounding soil. Mineral ions, however, are more abundant inside plants than they're in soil. Which of the method is most likely to allow uptake of the mineral ions? Okay. So uptake is a keyword here, and then abundant inside and outside of the plant. So let's just draw a little thing here. That's the root, okay? And that's the soil, okay? So soil has definitely, soil will definitely have lower minerals than plants, right? Because plants need to get lots of mineral from the soil, right? So at the root, plant tends to have more minerals inside than the outside, right? Because they are always intaking minerals, correct? So since you have this low concentration gradient established here and high inside here, is that clear? So based on what we learned from a couple of slides earlier, concentration gradient always travels from high to low, correct? However, in this case, we cannot lose minerals from the plants to the outside, right? High to low. That will be super detrimental for plants' longevity, correct? So what you have to do is you have to intake from the low minerals to the high state, which is inside of the plant cells. Is that clear? Okay. So the keyword is plant must uptake minerals from the surrounding soils. Then it kind of implies that we need to go against the concentration gradient, which means that we have to use energy for this to happen. That's why we have active transport will be the right answer for this case. Okay, so that's everything. That's all the slide I prepared so far. I hope you like this presentation, and this is pretty much how the class is gonna look like during the winter boot camp program. So if you like this demo, then I'll see you guys in the boot camp series. See you guys later. Bye bye.